Dear medical practitioners and allied practitioners, from myself, Marika, a very warm welcome to each and every one of you. I thank you for taking the time out to attend this event, and I trust it's going to be of great value to you all. This is our very first online collaboration with the wonderful team of Lancet Laboratories, and I'm hoping this will be the first of many more such online events and collaborations in bringing valuable information and sharing knowledge and expertise brought to you by the Lancet pathologists. You will note there will be a CPD application link in the chat box for your ease of reference. It is important to note that you will have to click on this link after the event to complete a CPD questionnaire in order to obtain your points for this evening's event. This link will also be shared after the event for your ease of reference. For those of you that are not too familiar with our company name, Richter Healthcare and Pharmaceuticals, you will probably know us much better through our very popular OTC products, such as our range of Salix products, as well as the Pure Skin range. You have just watched a short video infomercial, which was just aired, and that is proudly sponsored by Richter. We are the proud sponsors of this event and hope that we can make many such more events possible for you. Should you have any questions or any specific topics of interest, please feel free to share them with myself or Robin from the Lancet team. Later on, there will be a slide at the end with our email details for you to contact. Any topics of interest that can help guide your practice, or where you feel there's some issues that needs to be addressed, or something that will be of particular interest to understand and learn more from. We are interested in all the topics that would be relevant to you. Just some practical information tips for this evening. Please note all attendees will be muted throughout the presentation to ensure minimal noise and disruption. There will be a 10 to 15 minute time allowance at the end of the presentation for questions and answers. You will be able to type that in that very same chat box that I referred to. We will then relay those to the speaker. Each question will be addressed individually. Our speaker this evening is clinical virologist Dr. Karen Richter. She's a pathologist with an enormous wealth of knowledge, which she so kindly and willingly shares with you all this evening. Dr. Richter has achieved numerous merits and awards throughout her academic student career, such as Junior Researcher of the Pretoria University Day Faculty and receiving a Merit Award for her master's degree. She's also an acclaimed extraordinary lecturer of the Department of Medical Virology at the Pretoria University. So without any further ado, I would like to hand over to Dr. Karen Richter.
Good evening. I must say this is quite a challenging topic in that we cannot really predict the future. But we have learned quite a lot from international waves and from our first and second wave. So what we'll try to do tonight is not tell you exactly when the wave will appear and how severe it will be. Not even data analysts and people who predict the future can do that, but we can discuss the factors that will make an impact on the wave and see how we can make a change. So there's a lot of uncertainty. It is multifactorial. So if you try to predict what will happen, there can be a curveball like what happened in our second wave, and there is many unknown drivers, and there might be some unknown drivers that we're not familiar with. But what we know, and what most experts will agree, and we might be wrong because we've been wrong multiple times before when we made predictions in this epidemic, but there is general consensus that we are likely to face a third wave in the late fall or winter months of this year. But what can we learn from the first and second wave, and to apply to the third wave? So here we look at the first wave and second wave, the new cases that were reported cases in dark blue and the deaths in red. And on the top then the different levels of lockdown that we were on during the different stages. So there's a few things we can learn if we look at the data from the first wave. And you don't really need an epidemiologist or a mathematician or somebody who predicts the future to see that generally there's about 3 months between the end of one wave and the start of the next. So if you just use that very, very simple calculation, we can say that we'll probably see a wave by May, peaking June, July this year. So that is not really difficult. But we've also seen that from the difference, for instance, in our provinces, that there wasn't always a 3 months difference between a peak in one province and the peak in another province, and the order of peaks in the provinces differ in our second wave. So there is factors that can make this period longer or shorter. And especially in the period where we are now where we're starting to vaccinate, and we'll probably be too late with our vaccination program to totally prevent the third wave. It is very important for us to change our behavior so that we can prolong this wave for more and more vaccines to take effect. So it's quite relevant that we discuss it at this time period. So other things that we can see from the history and this graph, as I'm part of COVID testing from the start, obviously that's something that we've really experienced firsthand. And we know that the amount of testing or the tests available can also have an impact on the amount on your curves that you see or your amount of cases that's notified. And we know that in the first wave, we had a very strict case definition for testing because we simply did not have enough tests available. We just couldn't source enough from overseas. It was in high demand. And we know that we missed quite a significant amount of cases in the first wave, but probably also in the second wave. In the second wave, we have enough testing, but we have rapid testing that's done in doctors' rooms, etc. That's not always notified. So we know that testing can influence the waves, and that we probably had underreporting in both curves. Also, if you look at the excess deaths, but we can look at other factors like the death rate, the hospitalization rate, the positivity rates, etc., to know if we're missing a lot or if it's similar. And the testing for the two waves probably didn't have such an impact that the difference will be due to differences in testing behavior alone. The other interesting thing we can see from the first wave versus the second wave, we can see in the first wave we were in lockdown level 3, but cases started to decline as restrictions were actually being eased and social and economic activity increased. So that didn't make sense. So just mitigation measures alone will not necessarily increase or decrease your case, and there's a lot of cases, and there will be a lot of other factors. In the second wave, for instance, the mitigation level 3 mitigation measures made a big difference and actually had a significant impact on the lowering of cases. The other thing that we know, and I will show the province peaks in a further slide, but we know that areas that were significantly affected in the first wave were less affected in the second wave and the other way around. So basically, if you were in a province that were less affected, or in an area in your province, or in a social grouping where there was no immunity from the first wave, you could have you had the risk of being more affected in the second wave. So the message there is that if in your social grouping or network you were not affected in the first and second wave, you're quite lucky, but you are then highest risk for being affected in the third wave because you will not have immunity in your mini circles. So good examples is in Gauteng. The first wave were more severe in Johannesburg, and the second wave more severe in Tshwane, and also in PE. When the second wave started, it was more in the middle class neighborhoods, and the same for Tshwane. The middle class traditional neighborhoods were more affected, where they were not affected in the first wave. The other thing that could not be predicted by anybody is this driver of resurgence in the second wave, that we just suddenly see this marked increase, and that is probably multifactorial, but we will discuss the drivers, and it's good to then know that we must prevent the drivers if we can in the next pandemic, that we do not see this steep curve again. So this is the NICD data for the weekly incidence of cases by province and epidemiological week. So you will see that, as you know, in the first wave, it started in the Western Cape, and you would then expect the Western Cape to be the first to have a resurgence, but that's not what happened. The Eastern Cape were actually first with the resurgence, which started mildly and then increased steeply. The Western Cape was second, then followed by KZN, and then only Gauteng, while in the first wave, Gauteng were way before KZN. So there was factors, multifactorial, and we'll address some of them, probably maybe not all of them, because some are unknown, but why we see these different patterns. But it's probably due to human behavior, but not only human behavior, also other factors like viral factors. But we know, so we're not sure what triggered this resurgence in the Eastern Cape if you look at how human behavior alone. But I know a colleague of mine said every day as they drove past the graveyard in that area, every week there was like an exponential growth of funerals without proper social distancing and mask wearing, and something like that can trigger an epidemic. We also know there was the tin roof incident in the Western Cape, which was a super spreader event, and 198 other pubs or restaurants were investigated or closed due to infringement. So something like that can trigger a wave. But the one best described, and the one I'll just quickly describe, is the Lito Rage in KZN that was attended by private school matrics after the matric exams, and which most probably triggered both peaks in KZN and Gauteng, as 2/3 of the attendees were from Gauteng. But it wasn't these super spreader events alone. It was also a combination of summer holidays, people traveling, going to different social networkings out of their normal bubbles, relaxing, etc. And that is why we needed a lockdown to decrease the cases. And further, which we will discuss later, and just to give you a heads up, is the changes in the virus, which kind of went together with increased viral spread in these populations, especially young people who is asymptomatic and mingle. It gave the virus the chance to mutate and become more transmissible, and as it became more transmissible, it circulated more and it could mutate and accumulate more and more mutations. So this whole combination of factors of our behavior, but also the virus changing and the seasons changing and our behavior or our social networks changed, it probably had a combination of factors. So we can try and prevent some of these and not have a repeat in the third wave. This special public health surveillance bulletin was released by NICD last week and specifically look at the age group younger than 19 in South Africa. So why is this age group? And I just thought I'll just put it out there because I think this is quite something we should be looking at our third wave. Why is this age group so important? Well, they're more than 1/3 of our population. They do generally not get severely ill, but they can spread the virus. Everybody's told them since the beginning of the pandemic they're not high risk. So they really don't see why they should adhere to protocols. They love socializing. They love socializing with many people. So this is a population that we should watch also for the third wave and make sure that we do not have nightclubs masquerading as restaurants, etc., where these populations can start a third wave, and that this should be policed. But interesting in this group is also highly linked to schools, and in this report from NICD they discuss the school closures and the peaks of the epidemic, and which is quite interesting. The red areas is then when the schools were closed, so you can see there's no consistent change in incidence trends associated with the timing of opening or closing of schools. In the first peak, we had a decrease in cases when the schools were closed. In the second peak, we had an increase in cases when the schools were closed. It is probably highly linked to the background circulation of virus in the population. We know that from international studies, but we also know in the first peak, the schools were closed during a time where there was very strict other lockdown measures, and this really mobile population could not really move around and spread the virus between each other. But in the summer holidays, we were on level 1, we all had COVID fatigue, we all thought we might have beaten this thing, and it was just so nice to relax. And it was summer, it was Christmas, families got together, and also, which is not in this report, but we know now from the super spreader event in Ballito, is the private schools actually closed before the public schools, or the matrics. They finished about 2 weeks ahead of them, and they had their rage, and the mutated virus was there. They were all together, and they basically were infected, went home to KZN and Gauteng mainly, infected the public school matrics who just finished 2 weeks after and wanted to party, and then infected, unfortunately, many family members and grandparents and uncles, etc., as it was Christmas. So it was really a unique situation, and we'll probably not have exactly this very long summer holiday over June, July, etc. But we must be aware of the fact that this highly mobile population, especially the older adolescents and young adults, are vectors of the disease. Younger than 14, most probably not from most of the studies, but this mobile population should be looked at, especially if there's a variant emerging. So in the second wave, they were more likely to have COVID if you're 15 to 19 years old, male, and be in KZN or the Western Cape, where people went on holiday. And the report says it's possibly related to behavioral changes, difference in testing patterns. We tested more of these teens after the rage event, and or through differences between successive waves. So this just shows, again, in this less 19 years and younger population, the different provinces, and just how the Belito rage there in orange and light brown significantly increased the cases in Gauteng and KZN especially. Generally, just these, although they can trigger an epidemic, their cases basically mirror what we see in the rest of the population. So, what can we expect from a third wave? We know now from what I've said that human behavior and super spreading events is something we can control, and that can definitely spark a second wave, and is something worldwide that we should be concerned about. And if you allow super spreader events, you give the virus more time to mutate, and as a viral variant is also more transmissible, it can also cause more transmission and super spreading, more super spreading. So it's kind of a vicious circle. What do we have to prevent or make our third wave less significant is the mitigation measures, which we cannot really get go of at the moment. We can change our behavior, and behavior change is quite closely linked to seasonality. I'm not going to discuss seasonality as such on its own, but we know that it looks like we probably are going to have 2 peaks per year in winter and summer, but seasonality is linked to behavior change coming into our winter, that we will be more in enclosed spaces with closed windows, less ventilation, etc., which can make the virus more transmissible. SARS-CoV-2 vaccination is our big rescue, but it will probably not be in time to significantly flatten the third wave or stop the third wave, but probably waves in future. And then I'm quickly going to discuss what we know about population immunity, a lot of unknowns there, but it may save us. We may have more population immunity than we think. We are eagerly awaiting studies of population immunity in our country, but it might also be that the changes in the new viral variants make population immunity void. So let's just look at the impact of human behavior and mitigation measures. And this is, I think, one of the most important messages of tonight. And if you do not remember anything else, I want to just remember the significance of super spreading events, so that none of us are guilty of being a super spreader or sending our teenagers to be a super spreader or being part of a super spreader event. And this is the first study was done, published in Science, done in China, but it's also shown in other studies that basically 10 to 15% of people account for 80% of secondary infections. So if you look at this picture here on your right on the screen, every dot is somebody who's infected with SARS-CoV-2 and the people they infected. So you can see that many people actually infect nobody else, but there's a few people that are guilty of infecting others. Only 10 to 15%. So ideally, if you have public measures to stop super spreading events, we should not see any super spreader events. We just say no gatherings, etc. And it should, but unfortunately, our society is structured that we do sometimes engage in higher risk behavior. And an example from the Northern Hemisphere is that restaurants account for 20% of future months' transmission if you open restaurants. So we know by opening restaurants we will have some transmission, but we all know we cannot simply not open restaurants at all. So we might have many super spreader events, but we must try and minimize, and all restaurants are not equal. So it's also not fair to restaurants to blame just then, because you do get restaurants that adhere to precautions that are nice and open with high ceilings, good ventilation, and do not pack to more than 50% of their capacity. But you also unfortunately get restaurants that do not adhere and can be super spreading events. The other reality is if they have new variants with higher transmission rates, we will likely increase the frequency and size of super spreading events. And that's exactly what we're seeing now. We know we have a variant here that is higher, that has higher transmission rates. So if we look at these two scenarios, and a person on the left who did not infect anybody else and a person on the right who's a super spreader, what is the difference between these people? So how can we prevent super spreader events? So this super spreader here on your right is typically somebody that's highly infectious, and that is typically somebody that is either pre-symptomatic, so in the 12 to 24 hours before the patient will develop symptoms, so this patient does not realize that he's infecting everybody else, or is mildly symptomatic and he thinks it's not significant. So typically, adolescent or adolescent to teen or young adult will be in this category. So this just shows the importance of if you were exposed to COVID that you cannot socialize even though you feel well, because you can be a super spreader, but you might not know that you are in the incubation period. Secondly, these high viral shedding are seen in certain individuals. So there's quite a broad variation, but we know that older adults and adolescents shed much more virus compared to children, especially children younger than 10. So children younger than 10 is not really seen as being super spreaders or the vectors that spread disease, but all the children, teenagers and older adults are. Immunosuppressed individuals and then recently also shown overweight individuals are typically shedding more virus and can be super spreaders. And then the person's behavior is really important. So they're not adhering to mask wearing. So the moment here on the left, if you put a mask on this person, he will not be a super spreader. But if you're not wearing a mask, you do not keep your distance when during conversations, even outside, if you close proximity for more than 15 minutes, it is a risk. And then typically, when you speak loudly, this is all well described in the literature, the quiet practice example with the singing, funerals where people are emotional and they do not think of social distancing, family gatherings, nightclubs, cafes, restaurants, and gym classes where vivid exercise and ventilation is not good, as published recently by the CDC. And lastly, super spreader events is typically in closed spaces, poor ventilation, think of winter, crowded places, and close contact settings, the 3 Cs. So by avoiding these, and making some changes in ventilation and the way we do things, we can really prevent a lot of super spreader events. The other thing we've learned about super spreader events, which I think is really important and we should apply in the next wave, is we used to do forward contact tracing. So you are infected and we ask you, who have you been in contact with in the last few days while you were probably infectious? And then we alert those contacts to please isolate and not be a super spreader, which is efficient to prevent further spread if they listen. But a better way of preventing more infections was started in China, and this is called cluster-focused backward contact tracing. We know now, we said that super spreader events is about 80% of new cases. So if you see somebody that's infected with COVID, it's highly likely that this person was infected during a super spreader event. So by asking, who do you think infected you, or where could you probably have been infected, and identifying super spreader events, you can go backwards in time and alert everybody else that was at that super spreader event that they also possible contacts and prevent further spread from them. So a good example is the Belito Rage, where all the people who attended the rage were asked to isolate for 10 days, or they didn't necessarily listen and it was maybe too late. But if you could do that in time and you identified the super spreader event, you can prevent transmission if people adhere to the guidance. The problem with just with cluster-focused backward contact tracing, it is quite labor-intensive, and you can usually only adopt it when the case numbers are already coming down. So it is difficult and labor-intensive if you are on the peak of your wave. But at the moment where we see a few cases of COVID, it will be something that we should do. So just looking ahead into the future, what is the risk periods for super spreader events? And I'm sure you heard in the media as well. We expect the third wave, we said maybe May, June, July. Risk for periods of super spreading events is definitely first the Easter weekend. So we cannot allow any super spreader events to be allowed. Doesn't mean you can't go on holiday. There is safe ways to go on holiday in your bubble, to the bush, to the mountains, be outside, do not mingle with too many people. But a super spreader event over April holiday can be the amplifier to push our wave earlier and higher. The public school holidays is a bit distant from the Easter weekend, as you can see there, and there is no, currently, it might change, but public school holidays during May or June. So the amplifying will be in April if you look at the public school holidays. The private school holidays, although it's much less people involved in those holidays, are quite more longer and closer to the Easter holiday. So one can see that a super spreader event in the Easter holidays can be amplified if there's family gatherings and other mini or other super spreader events during the public school holidays, and this can definitely trigger our wave. So basically the message for tonight is whatever we do in April will have an effect on what happens in May, June and July. And although we hope to have some vaccines on board in April, we will definitely not have herd immunity by the end of April. So it is very important that we behave in April for one more time. We all want to go out and see lots of people, but that this can have a significant impact on our winter and our wave. So in winter time, as I said, not possibly seasonal, but the problem with winter is we're inside, we're in closed spaces, and this can also amplify our behavior linked to winter can amplify the wave in June, July. So we cannot let our guard down. We must prevent super spreading events. But is there any good news? Firstly, let's look at what we know about our immunity. And unfortunately, as I've said, there's many unknowns, but we know that most people develop an immune response within the first few weeks after an infection or after vaccination, usually 2 weeks after the last dose if it was a 2-dose schedule, or 1 dose if it was a 1-dose schedule. And we know if we test IgG against SARS-CoV-2 in the lab, it is reliable from about day 14. We also know from numerous studies that IgG antibodies may wane over time, but there's good news. We know that our body has something like memory B cells that can mount a really good anamnestic response with high affinity IgG antibodies when we're re-exposed. So even though our IgG antibodies may go down over time, when we are exposed to the virus again, we will have a good response. How long will immunity from natural infection or vaccine-induced infection last? Probably at least 8 to 9 months, but it may be longer. For other coronaviruses, we know that it declines over time. If we talk about population or herd immunity, the estimates used to be about 67% of our population that needs to be immune, but there is talk that it might actually need to be higher. So we're not sure exactly what we're aiming for, but it is at least 67% or higher. The bad news is that most global seroprevalence studies suggest that we're about less than 10% of that immunity at the moment globally. So how does our immune system react to SARS-CoV-2 if we think of antibodies? And I think this is important just to understand what population immunity means. So when we're exposed, and this is very, very basic immunology, the immune system is really complex and you cannot really explain it in a slide or two, but I thought this is really basic, and for those that are immunology purists, I ask forgiveness if this is not detailed enough. But if you're exposed basically to the virus, a white blood cell like a macrophage will engulf or phagocytose this virus and present antigens, which is protein parts of the virus, to the immune system. So in vaccination, it will be the spike protein that's depicted there. In natural infection, that's also really an important protein. But remember, there's other proteins as well, like the membrane and the nucleocapsid, and that can also trigger the immune response, especially the cellular immune response, which we'll discuss. So basically, these antigens get presented to your immune response, and your T helper cells, which is basically mainly CD4 cells, will then secrete signals, which is cytokines, to your B cell to please manufacture antibodies against this virus, and the B cell turning into a plasma cell will produce antibodies. If there's re-exposure to this virus and it's similar, this antibody can then actually neutralize this virus before you get infection, and that is first prize. But if your antibodies wane over time, there is memory B cells, as I've said, and they can actually remount a very good secondary IgG response. If the virus changed significantly, these antibodies might not recognize or fully neutralize the virus, and that is our concern with viral variants, because antibodies are quite specific for proteins, as example, the spike protein. But fortunately, all is not lost. To mention then, antibodies is the thing that we can easily test in the lab, and that is what you mainly test. It's a blood test, and it's affordable and cheap. But there's a vast part of the immune system that we cannot test on a routine basis, and it's quite expensive to test. And we must remember that, so that's only one snapshot of a whole part, other part of immune response. So not having antibodies does not necessarily equate to not being protected. So the other important part of the immune system is the T helper cells, or the cell-mediated immunity. So the T helper cell will signal cytokines to the cytotoxic T cell, which is usually a CD8 cell. And this cytotoxic CD8 cell will stimulate cells that can kill virus-infected cells. So these T cells are also a very important part of our immune system, and there's a lot of discussion how vaccines can also stimulate this part of the immune response. So why am I discussing this? So that you know that when we talk about seroprevalence studies, we're only testing antibodies and a subset of the immune response, and there is cell-mediated immunity that is also important. So antibodies is the one we test readily. It can block the virus from infecting cells and destroy it. On re-exposure, we have immune memory. It can reduce transmission by preventing infection. But if you have emerging variants that can escape or evade this antibody response, vaccines may be less effective, or a previous infection with the old variant might not protect fully against a reinfection against a new variant. But fortunately, we have cell-mediated immunity. Cell-mediated immunity do not necessarily prevent infection, but it's very important to prevent moderate to severe disease. And as you know, with vaccines, we are talking about, can a vaccine prevent moderate or severe disease? And that's mainly the cell-mediated immunity at play there. It can, and that's not always known, but it can reduce transmission, not totally, but by restricting circulating virus and having less virus, you can reduce transmission, although it would not necessarily totally stop transmission. So that is also good news. And because it targets many different viral proteins, it's more resistant to threats posed by emerging variants. And something else that's been discussed in the literature, but it's quite conflicting, so the jury's out on this one, but there is discussion that if you've been exposed reasonably recently to other coronaviruses, that your T cells might actually recognize some of those proteins that are shared between these viruses, and that you might have some kind of pre-existing immunity or cross-reactive immunity against COVID, and that might be an explanation why some people do not get infected with SARS-CoV-2. But as I say, conflicting evidence, and I just put it out there, but we're not 100% sure what that means. Okay, so the big question is, what is our seroprevalence in the different countries? Now, unfortunately, there's not a lot of peer review articles, and if you see peer review articles that's published in reputable journals now, that seroprevalence studies are usually about 6 to 8 months old, and we really want to know what's happening now. So I don't always use a non-peer reviewed data, but for this talk, I can refer you to the Sero Tracker website, which is more real time. It does, however, have national studies in many countries, as you can see in the blue dots, but there's also regional studies, local studies, like the one we will discuss from South Africa. There's actually two, but we'll only discuss, or we'll discuss two, but there's the one which is part of this website. And there is, so this data isn't perfect. There is a risk of bias. It includes local, sub-local studies. It includes mainly blood donor or residual sera, so meaning you had this serum in your lab from testing something else and you used it. So it's not necessarily a representative sample that can be extrapolated to the population. So we are eagerly awaiting results of more representative data, and studies are ongoing. But what this website shows is, and as I say, it's only one study from South Africa, but that we, in this population that was tested in Cape Town, we had a 40% seroprevalence rate. And if you compare that to many of the other countries, that is quite high. So maybe we get lucky and we have some kind of community population immunity, but we're not at 67% yet, but there might be something. But as I say, this is biased data, and we at least we're not at the bottom. So the studies that we have is, the first one was a study that was done at the end of this first peak, as you can see here at the bottom, when the cases was declining in July last year. So it was serum from women attending antenatal clinics and people living with HIV in public sector clinics in Cape Town. And they tested this serum randomly for SARS-CoV-2. And 40% of these samples had IgG antibodies. It varied significantly between districts, from a third to almost half, and females had higher antibody titers than men. But that, at the time, they hypothesized that this high level of community infection contributed to the observed decline in the epidemiological curve at that time of the third wave, when we were actually easing travel and other restrictions. The second study that was released in January, from January this year, which was released recently, which was done at the time when the second peak was decreasing in January, was done among serum that from healthy blood donors from the South African National Blood Transfusion Service. Only in 4 provinces. The Eastern Cape had a 2/3 positivity rate in that population. So it is again debatable how representative that is of the total population, but it's interesting to compare provinces, and also in brackets to compare it to the officially clinically confirmed cases. KZN and the Free State almost half, just above and just below half of patients that donated blood had antibodies, and in Northern Cape a third. So that is exciting that some populations at least have high rates. Also in the study, interesting that there was no evidence that the seroprevalence differed by age, but in the Black population, there was a 3 to 5 times higher IgG versus the non-Black population in this blood donor group. And in the conclusion, they felt that was a surrogate marker for your economic circumstances. And at lower socioeconomical circumstances exposed you to being more infected with COVID. Last data set is just data from our lab, which is also definitely not representative. There's bias. It's people who actually come to the lab to want their antibodies tested for different reasons. So I don't want you to read too much into this, but what is interesting is also at least there's a trend over time from August, when SAHPRA approved these tests, until February, that we went from a 1 in 5 positivity rate to a 1 in 3. So at least we are seeing more people with antibodies. So now with this good news that there might actually be population immunity, nature threw us a curveball, and that is genetic variants. So although we have IgG, we're not 100% sure how effective this IgG will be against reinfection with new variants, how effective our vaccines will be, and whether our T-cell immunity will save us or not. So what do we know about genetic variants? I'm only going to discuss the one in South Africa because this is relevant for the talk tonight, although we know there is other variants and lineages worldwide. So in this article that was published by the CRISPR group in KZN and in ICD and UCT, what is important here, this is the months when they did sequencing, and they saw that there was an accumulation of mutations in the virus from about September 2020, and the first in yellow cases of this new lineage 501v2 was seen from about October in yellow, and you can see how we just by November and December, or November on this slide, totally replaced all other lineages and it became the dominant strain. This is from the same article, just a map. So by doing phylogeographic analysis, it was suggested that this variant emerged in the Nelson Mandela area in the Eastern Cape in August somewhere, and then first spread to the Garden Route. So now thinking of those graphs of the provinces and the second wave, we can see how this is probably the most significant reason why the Eastern Cape had this early peak first because of this variant, and then the Western Cape Cape and the Garden route, and then from there spread to the rest of the Eastern Cape, Cape Town itself, and KZN. The other study or data from NICD is also on the same lineage from, so there was sequencing done from samples from Gauteng, and what is important here is this in the pink purple, that is in November and December, how the lineage spread to Gauteng in November and basically replaced all other lineages. 95% that was circulating December in Gauteng was already this lineage. So if you got infected with COVID in December or January, February, it was most probably the new lineage. This just in a reminder of that super spreading event and the lineages with the different provinces, and you can see how the Eastern Cape, followed by the Western Cape, followed by KZN, where we then had at that moment a super spreading event with a highly transmissible virus, and that probably fueled our whole third wave. So what is important about this mutation, just a little bit of virology, one slide. So this is the spike protein of COVID, and here we have the receptor binding domain, or RBD, which is the important domain because this is where the virus binds to the ACE receptor on the human cell. And changes in this receptor can make the virus more transmissible if it can bind more effectively. So there are three mutations that's important that you've probably heard about, N501Y, E484K, do have an effect on the ACE receptor binding, and the K417N also enhance binding, which can, so the virus binds with a higher affinity, so binds better and is then more transmissible. So these mutations has also been seen in the UK and the Brazilian and the US strains or lineages. Yeah. So further on, these mutations can then also have an effect because there's a 3D change in this area. Our antibodies that were from the old strain might not work as well, and these mutations are actually shown to confer or have abolish interactions with neutralizing antibodies to different classes. So this may unfortunately contribute to immune evasion or escape. And then devastating in January, this was confirmed by this article published by the South African National Blood Service and other multiple universities and NICD, where they actually then showed what was feared, that there was complete escape from 3 classes of therapeutically relevant monoclonal antibodies and a substantial or a complete escape from neutralizing antibodies in patients with convalescent plasma. There is immune escape evasion, and they said then in this article there's the prospect of reinfection with antigenically distinct variants, and it may foreshadow reduced efficacy of current spike-based vaccines, and how right they were. So this caused AstraZeneca to bite the dust, and now everybody to scramble for other vaccines that might work, have clear data if it might work or not, and to actually decide which vaccines will actually have a significant effect against the variant that is the dominant variant in our country. So what is the impact of SARS-CoV-2 vaccination on the wave? I only have one table here with data of the vaccines that we are most likely to see in our country and that actually has data of efficacy or no efficacy events against this lineage. So there's many other vaccines, but like Moderna will probably not get because it's too expensive and it will come too late. And the other vaccines, the Sputnik and the Indian vaccines, do not have data against our variant. So what is important in this table, if you look at the AstraZeneca vaccine, the reason why we're not using it and it's in a warehouse is because of the 10% efficacy shown, unfortunately, against our variant, although there was very good efficacy against the old variants. So if you look at the Johnson & Johnson and Novavax vaccines, there's actually trials were ongoing in South Africa when this virus was circulating. So we actually have data of a 57% and 60 and almost 50% efficacy of these two vaccines in the HIV versus the HIV negative versus a mixed population. So there is some efficacy shown with Novavax and Johnson & Johnson, although Novavax is not approved yet. For Pfizer-BioNTech, there's lab studies that were done in a Petri dish that showed that you need a bit more of the antibodies, increase, but not, it still neutralizes, you just need more. So you just need enough. So this is also why the Pfizer vaccine is a possibility to be used in our country. So there is some vaccines with efficacy and some vaccines that we do not have data that might not work against our vaccines, against our variant. So it's talking about vaccines and impact. If you look at the slide, there is different communication from different people when we will have vaccines on board. So the aim is by mid-April to have at least 5.5 million or 500,000 healthcare workers vaccinated. That would not have a significant impact on the third wave, but definitely for healthcare workers. Another talk is about 6 million vaccines on board by end of June. That's about 10% of our population. So again, what will happen in April with our behavior, etc., is unfortunately much more important because we will probably not have vaccines that can significantly impact our third wave. I hope I'm wrong and we get many vaccines and a good rollout, but at the moment, we cannot rely on the vaccines to prevent our third wave, and that is just most probably the reality. So what is the possible scenarios? So the worst case scenario that might happen with the third wave is that our immunity is waning or is incomplete and we get variants that we can, our immune system does not recognize, and we actually have a lower population immunity than these other studies may suggest. Our behavior allow the virus to circulate unchecked, and we get more and more highly transmissible viral variants that escape natural and vaccine-induced immunity, and probably worst case scenario mutate to cause severe disease. Our vaccination rollout is delayed or the uptake is low, people do not want to get vaccinated, and our vaccines are not effective as we want against the new viral variants. And part of this worst case scenario is us having COVID fatigue. We are selfish and indifferent and decide whatever we do, we're low risk, we do not care about what happens to other people, and we do not adhere to mitigation measures. And our government do not enforce or the mitigation measures, especially during the Easter holidays. We have super spreading events. This is worst case scenario. Then, and Discovery was brave enough to make a prediction, we will have a third wave that's much worse than the first and second wave. And Discovery predicted last week that we can have an additional 92,500 COVID deaths by December. So this is if we behave like this. But there's hope on the horizon. The best case scenario is that we have actually a significant and effective population immunity, and our T cells come and save us with long-term cross-variant immunity. Our cell-mediated immunity works. We have less virus circulation and limited emergence of new viral variants because there's less circulation, and the viral variants have a decreasing virulence and pathogenicity. We have sufficient coverage with effective vaccines. We delayed the wave and we have time to vaccinate to prevent severe disease and death, and some of the vaccines also decrease transmission either via cell-mediated or antibody immunity. And we adhere to mitigation measures for a little while longer, especially focusing on not causing super spreader events or being part of a super spreading event. Then we'll probably have a third and maybe a fourth wave in the summer, but with time, as our immunity increases and the virus gets less virulent, the virus will become endemic, meaning it will circulate like all other coronaviruses, the seasonal corona or the normal coronaviruses. And we might have resurgences now and then regionally or seasonally, and it will basically called mild to moderate disease because everybody will have some kind of immunity against severe disease. Children will get the virus when they're young because everybody else will have immunity, so they'll get there when they're younger than 6. It will be like a childhood virus where they won't get severe illness, and we'll learn to live with the virus, quite similar to other coronaviruses, but also really what happened with swine flu. Swine flu is not really an entity that we're worried about 10 and 11 years later. But the message is that individually we cannot let our guard down now, and we can only change our behavior. So I ask you to not be a super spreader or avoid potential super spreading events, and choose no or low risk alternatives, and also let your family members and your teenagers and your young adults in your home be super spreaders or attend super spreading events. And it's easy to identify these. And don't be picky. If you get the opportunity to get the vaccine, just get whichever vaccine are offered to you at the time that it's presented, so that we can get as many vaccines on board as quickly as possible. Now, to just conclude, what will happen after these waves? The Nature article had asked immunologists and epidemiologists and scientists all over the world what they predict will happen. And the majority, almost 90%, said that they think COVID-19 is here to stay. But what it means isn't as dire, and it really relies on our vaccination efforts. And basically, just in short, if we have vaccines and even if they escape the immune system, we can work around that. We can give boosters if our immunity wanes. We can change our vaccine formulation regularly. So that is not an issue. If the vaccines can totally block transmission, and we can vaccinate enough people, we will probably eliminate COVID in some regions, similar to measles that you do not, for instance, really found endemic in the Americas, and it will just be reintroduced from areas like possibly Africa where it is not eliminated. So this is a possible scenario. More likely scenario, though, is that we cannot really vaccinate wide enough globally, but we do get some kind of immunity so that the virus will just basically circulate as a mild to moderate disease. Especially as we know, it seems like our vaccines can prevent severe illness. If the vaccines cannot prevent severe illness, we will continue to have severe disease, but fortunately, this does not look like it will be the case. And lastly, there is some talk about animal reservoirs, and if the virus can cross the animal barrier again, we will maybe see future outbreaks, but this is not really our concern at the moment. The good news is that after these initial peaks, there is light on the horizon that we will probably learn to live with COVID and have mainly mild to moderate disease. So to conclude, South Africa will probably, unfortunately, have a third wave, and I will be very happy if I'm wrong, and it will probably be late autumn, winter, with the inception in May to June, with a peak in June to July. But it is difficult to predict because it is so multifactorial, and it might be a curveball coming our way, which we did not think of. Our vaccination program will probably be too delayed now to have a significant impact on our third wave, but definitely on fourth and subsequent waves. Social networks least affected in the first and second wave will be most at risk because they have a bigger population of at-risk people. Worst case scenario is that we have a significant worst wave because of escape variants and super spreading events and our immunity that is incomplete. But best case scenario is that we do have some level of natural immunity from our first and second waves and the few people, the lucky ones who were vaccinated. And our variants do not mutate much further, and we are responsible and we limit super spreading events, and we then have a milder third wave. Thank you.
Dr. Richter, thank you so much. That was a very insightful, knowledgeable presentation that you shared with us. If I can just relay one of the questions that has popped up in the chat box. Stella is asking, what would the reason be? Are there specific reasons for testing negative, even though patients are positive?
I assume that's with the PCR test. So there is multiple reasons. You must look at the natural disease process and where the patient is. So if you were exposed and you test too early, you're still in the window period, you might have a negative test, although you will start shedding virus the next day or the day after and can still infect people. A second scenario is when the sample is not taken properly so you do not have proper cells, then you will definitely get a low, can get a false negative result. Thirdly, so the test is most sensitive in the first 3 to 5 days after onset of symptoms. So then a false positive test is unlikely if you take a proper sample. But late in the disease process, especially in the second week of illness, we know that there's more virus in the lungs than in the throat, and it's an immune-mediated process. So then there is actually less virus in the throat. So then a throat swab or a nose swab might not pick up virus, but if you take sample from the lower respiratory tract or the lungs where there is actually virus at that stage, you will diagnose the infection. So there's a variety of factors that can cause that, and if a negative test does not fit the clinical picture, it is important to repeat the test or repeat it from a different site. For instance, if there's symptoms in the lower respiratory tract, from the lower respiratory tract. There's also some doctors that prefer to in patients with who mainly present with diarrhea, feel that throat swabs are not as sensitive, and they prefer to send a rectal swab in a subset of patients, for instance.
Thank you, Doctor. I see that Stella did hit the like mark there, so I think that suffices to her question. There's no other questions at this stage, so I would like to just once again thank Dr. Richter for taking the time in preparing this wonderful talk, and also just thanking all the attendees for attending this evening with us. We will keep you posted with regards to future events. Also, I just want to make mention, you will see there's a slide on the screen now with the details of Robin from the Lancet team, as well as my own details from Righter's side. We will be sending hampers to the first 120 that registered, just as a thank you for attending this presentation. Also, I want to just ask you the link that I mentioned earlier on that is in the chat box still. If you can please just click onto that link and just complete the few questions in the questionnaire to obtain your CPD points for this event. Then lastly, I want to mention that this whole talk will be available as a recording on the Lancet website. So if you want to recommend the talk, or if you want to refer back and just for your own knowledge, just go and view it again, or just tell a fellow colleague if they missed out on the event, then they can go onto the Lancet website and then they can go and view the whole recording there. The CPD link will also be available there to still then complete the questionnaire after the recording, and then you will still obtain your CPD points. So thank you very much for attending. It was a lovely evening.